Bismillahirrahmanirrahim. Can you all see my screen?
Yes, sir.
So today we are going to have a quick review of the cell signaling and how cell signaling is involved in development. And we'll pick a couple of examples just to highlight the significance of intracellular, intercellular communication or cell signaling in the formation of a organ and tissues. As you all know that in multicellular organisms, there are different specialized cell types are formed which eventually contribute to the formation of organs. And the complexity of human organs or organs in a multicellular organism is that they are composed of numerous type of tissues. Not just one kind of cell type is there. For example, if we talk about the limb development, we do have muscle cells, cartilage, bone, etc. And all of them come together during the process of morphogenesis to form a perfect organ which is able to perform its function. Another beautiful example is human eye. And if we try to look at the different kinds of tissues there, we have corneal tissue, which is responsible for the transmission of the light. Then we have the lens, and the diameter is controlled by the muscle tissue. And finally, we have the neural retina. So now you have corneal tissue, you have lens, you have muscles, you have neural retina, and all of them, they come together to form a human eye, a functional human eye. Any of the tissues, any of the cell types which I just mentioned, if they become defective, you know, you may have the morphological structure there, but the function may be gone. So there are two concepts in terms of development of organs when we talk about the cell signaling. Those two concepts are basically the induction and competence. Induction, as the word literary meaning is, to induce, and then competence here refers to that if I receive a signal which is inducing me, I need to have factors and I need to have ability or competence to respond to that particular signal. And in this context, we have example of optic vesicle, optic cup in the formation of lens and eye structure in frog. So, as I said, if there is a cell type or a tissue which is emanating or sending a signal, that signal is to be perceived by neighboring cells or tissue, and the one which is sending signal is basically the inducer, and one which is receiving that particular signal is the responder, the one who is to respond to a specific signal which is going to induce some changes in this second tissue or cell type. Now, this responder need to be competent. Competent means it should have the factors which not only receive the signal, but also generate an appropriate response to a specific signal which is coming. There can be close range interactions, and there can be long distance interactions as well. So for example, in case of close range interactions, we talk about, two neighboring cells or cells in close vicinity. And then long distance interactions can be, one signal, one cell type at a long distance or far away from the inducer cells. When we look at the formation of eye structure in Xenopus, which have been used very intelligently to understand the concepts of induction and competence and the associated cell signaling, is so we have the optic vesicle. So this structure is this one is the optic vesicle in Xenopus. Now, this optic vesicle is going to make the cell fate is retina. So it's presumptive retina in Xenopus. If you now transplant this tissue, so this optic vesicle, and you bring it at a different location or altogether in a different environment. So here, this optic vesicle is not able to induce changes in this ectodermal cells. This optic vesicle, it sends signals, molecular signals, which tells ectodermal region in the head region, which is this one. So this one or this one. So if you remove the optic vesicle from here, you can see the ectodermal cells, they are not invaginating. They are not, going into the fate or into a form which eventually is going to produce the lens. So it means this optic vesicle, the cells here, they are sending some signal which is perceived by the ectodermal cells here. And as a consequence, these cells then invaginate and contribute to formation of lens. So in this case, optic vesicle is the inducer, the one which is sending an inductive signal, and the ectodermal cells are basically the responders, the ones which respond, and they are competent enough because they respond in the form of a specific conformational or morphological change, which is validated or supported by once we remove this optic vesicle. If we remove this optic vesicle, transplant it in altogether a different position, now the signal is there. Induction signal is coming from vesicle, but now these cells here, they are not responding. They are not responding because they are simply not competent to respond to the signal which is coming from optic vesicle. Is it clear? The concept of induction and competence?
Sir, I have a question.
Yeah.
That the optic vesicle will be sending signal, will be inducing even in other regions as well, right? It's just that the ectodermal cells are not competent.
Yeah. So ectodermal cells in this head region, this one, these ones are responder cells. They are competent, they will respond to the signal. But in this region, the ectodermal cells are simply not competent. That's why they don't respond in the way the ectodermal cells in the head responds. Yeah. So what kind of changes or what kind of behavior could be involved when we send some signals to the neighboring or the adjacent tissues, etc. So it can simply result in change of shape, like here you saw. It may influence the rate of cell division. It may even induce change in cell fate. So look here, you were simple ectodermal cells, just like your neighboring cells. These are the ectodermal cells. But as a result of induction, as a result of signals from optic vesicle, these cells are going to attain a specific fate, which is going to result in the form of a lens formation for the eye. So you have to, as I said in my last lecture, when we learn developmental biology, or when we think in terms of development, we have to think in terms of, a cascade of changes which may result as a consequence of a specific trigger. And here the trigger is basically a kind of signal coming from optic vesicle. And then that signal is received by ectodermal cells here. These ectodermal cells are going to definitely generate a corresponding response, which we will see in the next slide. And what those responses are, so we'll see the molecular changes, how this lens formation takes place. Yeah, Abdullah?
So, sir, this is just the beginning of morphogenesis, right?
Yeah, this is morphogenesis, because this is just the getting into a form. A specific form is being established. This is just the beginning. Pattern is being laid down. Yeah.
So something has already told those cells, those ectoderm cells near the optic nerve, that they have to differentiate in a way in order to accept or process the signal from the optic nerve. There has already been some signaling which has caused those cells to differentiate in a certain way.
Yeah. So you remember we did talk about formation of the germ layers. So ectoderm, when we say this is ectoderm, it means these cells have undergone certain kind of cell signaling, some signaling associated with the environment they are in, the milieu they are in, and they have attained the ectodermal fate. Now, when they will receive this particular induction signal, these ectodermal cells, they are going to respond in a specific manner. You're right, there is some kind of already cell fate determination, and that cell fate is a very loose determination, which is just the ectodermal fate. Clear?
But sir, the other ectoderm cells are not responding to the same signal. So only the cells which are near the optic nerve, they have been in that certain differentiation.
Yeah, only the ectodermal cells in the head region will respond, not in this region, in the trunk. Which means there is a pattern already laid down. A pattern is laid down already while we are having three different germ layers. Yeah, there was another question, I think, Fariha, or?
Yes, sir. So I was just wondering if it's possible for the signaling molecules to move from this part, from the trunk area, somehow to the, to those cells which are competent to respond. Like, is there a possibility for that?
From which part? From here?
Yes, from here. So since we have the optic vesicle here, and
Yeah.
if we have signals here as well. So
Yeah.
is there any possibility that the signaling molecules will move from here to the head part, where we have, like, where the ectodermal cells are actually responding to these molecules?
So you are saying signal from here can go all the way there.
So basically, in the, like you mentioned earlier, that there are some signaling molecules which, which results in the long distant interactions, or signaling.
Yeah. So when last time we talked about, just a very preliminary paracrine factors, etc. You should know that in cell signaling, there is always a threshold concentration. Threshold concentration is involved. And also there are different kinds of signals. For example, the paracrines, they can diffuse. Now, when they diffuse, let's say if it is a big cavity here, there's no hindrance, so these signals may come there, but if they are below a certain threshold concentration, these cells won't, those signals won't matter for these ectodermal cells, even if they diffuse. However, since this is here in the close vicinity, these signals will immediately, in a big concentration, will come here, and we expect that this ectodermal cells, these ones, they should behave this way because they are receiving the same concentration, threshold concentration, which is causing changes here. Did I answer your question?
Yes, sir. Thank you.
So now let's look at the molecular level. What are the induction signals and what are the factors which result in competence, which make responder cells competent to generate specific response? So competence is an actively acquired state. And in this particular example where we are saying, optic vesicle generates specific signal and ectodermal cells responds because they are competent. They are competent because they have a specific transcription factor, which is also referred to as master regulators, a master regulator of eye development, PAC6. So PAC6 is a transcription factor which confers this competence to the ectodermal cells in the head region. When the signal comes in, and the responder cells, they respond by activating PAC6. So PAC6 is the competence factor here. How do we know that PAC6 is the competence factor? The homologue of PAC6 in fruit fly is called ILS. When I use the word master regulator of eye development, master regulators are key developmental genes which, when we ectopically express, they can result in formation of, let's say, eye or wing or a specific organ. Okay. In PAC6 alone or ILS alone, let's say ILS in flies, when we ectopically express in the head region or wing, in the leg regions or wings, you all may remember I used to show this beautiful picture from Walter Gehring's lab where they could generate eyes on the leg or eyes on the wings. That is due to ILS, ectopic expression of PAC6. So how do we know that this is the competence factor? You can simply use homologous recombination and you can generate a homozygous mutant, mouse mutant, which is deficient for PAC6. So I'll ask you all today, your assignment is go back and read about homologous recombination. To generate knockout mouse. Okay, so you are going to read about, you're going to understand the tool of homologous recombination, which is employed to generate knockout mouse. Okay, so when you generate knockout mouse, the phenotype of mouse is, look, this is the wild type. This is the eye structure developing, the mouth, lips, the brain, etc. Now, just the deletion or knockout of the PAX6, eye is gone. No more eye. The mouth is deformed. And if we take slices of the brain, what we see that the forebrain and the formation of the brain is drastically impacted. There's a severe defect, and these mouse, they die at early stage. Now, as I said, you have to, once you generate a knockout mouse, and here now this tool is extremely important in this course, and in subsequent lectures also you will see. You can also not only read about homologous recombination, but also read about CRISPR mediated knockout. Okay. Nowadays, instead of going through pains of homologous recombination, creating a knockout mouse using homologous recombination used to be very tedious job. And it used to be a complete PhD project. And if you are not lucky, by the end of your PhD, you realize my mouse is not showing any phenotype because the gene may not be that important. And then the characterization of that knockout mouse was an independent PhD project where a follow-up student will characterize. Now, with the help of CRISPR, all this has become very fast. Read about CRISPR-mediated knockout as well as homologous recombination. Why I want you to learn all this? Because if we are going to learn about development, if we are going to understand developmental biology, we have to understand correct use of these tools. We have to understand how am I going to use knockout mouse, CRISPR-mediated knockout, RNAi-mediated knockdown, what we call the genomic rescue experiments, and so on. Unless we don't understand the functional genomics part of the molecular biology, we are not going to understand and enjoy the concept of developmental biology. So let's say we generate, our hypothesis is, and pay attention to everything I'm talking about. Our hypothesis is that PAX6 is a competence factor. In order to prove our hypothesis, we generate a mutant, a CRISPR mutant or homologous recombination, knockout mouse, etc. Now, once you have this mutation, we have the phenotype. This shows, PAX6, if I target PAX6, that results in a very specific phenotype. But I have to validate, I have to prove that indeed PAX6 is gone. Just homologous recombination or CRISPR or any other tool which you are trying to use to generate non-functional PAX6, they may target somewhere else as well, although they are very, very specific tools. But there are always exceptions where we have off-target effect. So in order to show that this phenotype is due to loss of PAX6, we can do in situ hybridization. In situ hybridization is basically you prepare tissue. In this case, for example, you have these brain slices or the mouse slices. If you're working in another organism, let's say flies, you will take larval imaginal discs or early embryo, okay, and then you use the probe which is labeled when we do in situ hybridization, this is like we used to use the dig label. These are fluorophores, and PAX6 or ILS, which is the homologue of which is fly homologue for PAX6, we will in imaginal disc or early embryo or these slices, we will hybridize them with this fluorophore containing probe. Now, wherever, as a comparison, we are going to use a wild type. So wherever PAX6 will be present, what we will see, for example, this staining, dark staining. This shows these are the places where PAX6 is expressed. Look, it is expressed right here in the brain and the eye regions. However, when we will see the same stage knockout, we'll see this signal is missing. So what we learn, what we see, that with the deletion or knockout of PAX6, the expression of PAX6 is missing. And this is the first experimental validation or correlation. You say, okay, I targeted PAX6, PAX6 expression is gone, and then I have corresponding phenotype. This phenotype is most likely due to this. You can also think of using RNAi and say, okay, instead of knockout, which can result in death of embryo, I can use, tissue-specific induction of RNAi signal and then reproduce what I see through knockout. That is now a different experimental tool you are using. And another thing is, once you have this phenotype, in genetics, what we do genomic rescue or functional rescue. So we will bring back the PAX6 or ILS back to the mutant and rescue the lethality and this phenotype. So all these are kind of the various tools which we use. Now, this shows that in the absence of PAX6, we don't see eye formation or head ectoderm is not able to. Now, what we are, hold on. What we have just seen is, we have seen that PAX6 is indeed the competence factor, but we haven't yet proven that, whether these changes are happening in optic vesicle or it's happening in head ectoderm. We have to design new experiments. We have to do new experiments. So we do, in order to further validate that, PAX6 is indeed the factor which makes the head or surface ectodermal cells competent to respond to signal coming from optic vesicle. This kind of experiment is going to show you the beautiful conclusions. So what you have optic vesicle cells, you have surface ectoderm, both are wild type, and you have lens induction. Okay, this is lens induction is taking place. Now you generate deletion of PAX6 specifically in the optic vesicle, and we call them such knockouts as conditional knockouts. Conditional knockouts are when you are knocking out a gene in a specific tissue, cell type. For example, only in heart, only in neurons. So this is conditional knockout. Now, you knock out only PAX6, so PAX6 minus minus means deletions in the optic vesicle. Surface ectoderm is normal, and you see lens formation is still taking place. Now, what you did next, you have optic vesicle, which is wild type for PAX6, but the surface ectoderm lacks PAX6. minus minus, no lens formation, nothing. And if you have both, minus minus, again nothing. So it means PAX6 is the competence factor which enables the surface ectoderm, head ectodermal cells to respond in a specific manner, which then result in morphological changes in the form of lens formation. Is it clear? Is it clear?
Yes, sir.
Yes, sir.
So now, is it just, PAX6? Is it, like one fine morning, optic vesicle sends a signal and um, head ectoderm responds by activating PAX6? It's not that simple, as Abdullah was asking question earlier. These changes take place much earlier. Development is a very, very precisely engineered process. It's not, random collision of factors and, events. A minor mistake, you can see, this is a minor mistake, just a one gene. Out of 20,000 genes, this is just a one gene. And look what consequences we have. So even before we arrive at the stage of PAX6, this optic vesicle induction signal and then competence factor PAX6 from head ectoderm, these changes result due to much earlier changes at the gastrulation stage or even before gastrulation stage. So Abdullah, answer to your question is coming now. So at early embryonic stage, when we are in the mid-gastrula stage, the presumptive pharyngeal endodermal cells, it's not yet, pharynx is not there at all. This is where you have the pharynx. But the presumptive, the cells whose fate is presumably the pharynx, and they come from endodermal origin, these cells, together with the heart-forming mesoderm, they send the signal, earlier signal, which we know, and maybe this could be maybe very prior to this stage. And this signal comes and tell the anterior neural plate that the cells at the ectoderm, they go in a depression mode, go like this. These cells then sends the signal in the form of FGF8 and BMP4. These signals are coming, this is basically the optic vesicle. So these are color coded. BMP4 is this, and FGF8. These two signals are coming from the optic vesicle. But who is inducing optic vesicle to send these signals? Even earlier cell types, pharyngeal endoderm and heart-forming mesoderms, they somehow resulted in these production of these signals. Once these signals are produced, they are received by the lens-forming ectoderm, and they start expressing two transcription factors, SOX2/3 and LMAF. And in this whole scenario here, this is the inducer. Optic vesicle is the inducer, which is sending signal in the form of FGF8 and BMP4, and lens-forming ectoderm, the head ectodermal cells, they are the responders, okay? And they respond by producing these transcription factors. Now, if you look at molecular level, what is happening, how PAX6 is contributing in this whole process? If you look at the genes here, they are, so this is the promoter region. Then you have the PAX6 binding sites, MAF binding sites. You have PDX, PBX, etc. So all this PAX6, SOX2, SOX3, LMAF, they work in a very, very specific coordination. It's the coordinated action of these transcription factors which produces changes which sets the stage for lens formation. If one of them, and they have done these experiments, if you create deletion in the DNA for, let's say, the PAX6 binding sites. So PAX6 is there, but its binding sites are missing. You will find such phenotypes. Although wild type PAX6 is there, but you're not able to execute response to this signal which is coming from inducer. So this cartoon is oversimplified version which I drew myself. Otherwise, it's just text. Otherwise, it's just, and we can see this in the form here as well. This is from the book. So look, this is mid-gastrula, early to mid-gastrula stage, which is depicted here. This is from your book. And here you have this pharyngeal endodermal cells signal. This is basically the signal. On the y-axis, if you relative capacity to induce. So this is pharyngeal endoderm, then the heart-forming mesoderm, and they set the stage or onset of optic vesicle is due to these signals. And then once you have optic vesicle formed, you have here then a response in the head ectoderm. And eventually you have, different tissues in the eye being formed. If you look at the same graph in the form of embryonic picture, so this is your mesoderm, dorsal mesoderm, this is endoderm, and this is your ectoderm. And this is the presumptive retina tissue, this one. This is your neuralized ectoderm being neuralized, this one. And if you see now, this mesoderm, dorsal mesoderm, this signal is coming, and that is going to result in neural plate formation. This neural plate formation then results in the formation of optic vesicle, and this optic vesicle sends signal to the ectoderm, head ectoderm, which results in the lens formation. And this neural plate is also, you can see the earliest brain or as a result of neurulation. Along with these morphological changes, what is important, you see transcription factors OTX2, then PAX6 is expressed in presumptive lens ectodermal cells, which are these ones, and then you have coordinated expression of OTX2, PAX6, SOX3, etc. All this is actually to highlight complex or intricacy of signaling events taking place, which we take for granted, let's say, in the eye formation. This eye is due to earlier events which have shaped the eye due to these signals. Now, it doesn't stop there. It does not stop there. once inducer, oh, sorry. So this was inducer, this was responder. Now we call this reciprocal induction. Once PAC6, SOX2, 3LMAF are induced, now the former responder, it becomes inducer, it starts sending signal, and the former inducer become a responder. We call this reciprocal induction. So if this does not take place, lens formation, eye structure will not be complete. Okay, so once the lens becomes inducer, now this lens sends specific signal, and that specific signal causes the optic vesicle, which was formerly the inducer, to even further go undergo morphological changes and take up shape of an optic cup. And this wall of optic cup then differentiate into two layers. One is pigmented retina and other is neural retina, which will be clear here in this picture, for example. You have neural ectoderm, you have the lens placard. This is the early optic cup. Now this you can see is the lens forming ectoderm. So here you can see the optic vesicle taking up shape of optic cup as the lens forming tissue is now sending the signal. And you have, due to this reciprocal induction, you can see there is a clear lens capsule there, lens vesicle there. And due to reciprocal induction, now you have neural retina and then the pigmented layer of retina, which are the two cell types which differentiate in response to reciprocal induction by lens. Now, this is going to summarize all what we have learned so far. We are at early gastrula stage, late gastrula stage. So at this particular stage where we have presumptive pharyngeal endoderm and the heart forming mesoderm, they start sending signals which result in lens ectoderm because the neural plate formation is a consequence of signal coming from these mesodermal and endodermal cells. This neural plate results in the formation of optic cup, and which results in optic vesicle. All this is due to signal which started from pharyngeal endoderm or heart forming mesoderm. And then all the subsequent changes, we have seen that PAC6 and SOX2, LMAF, etc., they are the ones which respond and result in lens formation. So here you have PAC6, SOX2, LMAF, etc. And on this side, you have signals coming from BMP4, bone morphogenetic protein 4, which are basically these signals. Okay, sorry, not here, here. Okay, so I take a break here. And what is time now?
10:48.
Yeah, let's take 10 minutes break, and then we continue with the kinds of signals and some classical experiments which showed induction and you know competence, etc. Is it okay? Let's take 10 minutes break.